This is a production of Cornell University.
So one of the great things about high-throughput plant phenotyping is, from the plant breeding side, is we've been thinking about high-throughput phenotyping for quite a few years. But plant breeders themselves have been doing phenotyping for the past 10,000 years, as they've tried to get plants to feed and clothe the world and provide food, fuel, and feed. Thankfully, not all plant breeders evolve at such a slow rate over the past 10,000 years, and we quickly have realized that there is a lot of tremendous technologies that can be applied to our field of plant breeding. And it's, in a sense, forcing us to think more broadly into computer science, engineering, robotics, and artificial intelligence. So this symposium, I feel, isn't just for plant sciences themselves, but it really is also an opportunity for us to reach out to other departments on campus and for them to see the value of our work and how they can fit into that plan. So our first talk today is going to be by Todd DeZwaan. He is from one of the best phenotyping companies in the world, and the title of his presentation is Phenotyping Solutions for Basic and Applied Research in Plant Biology and Agriculture. All right, Tom?
Great. All right. Well, thank you very much. And thank you, Alina, for the invitation. I think we started this conversation a couple of months ago, or 6 weeks or so ago, and it's really nice to be able to follow it up so quickly. I really appreciate that speed. Speed is so important in this area of endeavor. So I'm going to be talking to you about phenotyping solutions for plant biology and how you can apply them to agricultural research applications. I'm coming at this from the perspective of today being the business development lead for a technology company that's developing solutions in this space. But my background is, prior to that, I spent 11 years working with Monsanto, and specifically my last role with Monsanto before I left the company in September was a strategy lead for a precision phenotyping system that they had developed, that they constructed in Research Triangle Park, and prior to that was working with Paradigm Genetics, and we were working on what we called the gene function factory in Arabidopsis between 2000 and 2005, where we were doing a lot of high throughput phenotyping in Arabidopsis. So while I'm pitching a product, if that's the right thing to say today, while I'm here representing LemnaTec, I also have a pretty extensive background in the application side of things. So I'll start out a little bit just echoing what Mike said about the history and talk a little bit about the challenges coming primarily from the genomic side of things. Won't talk too much about the environment, but of course G by E is crucial to anything that you do in the phenotyping space. And then I'll shift from there and talk a little bit about technologies that we're developing at LemnaTec and how they address some of the solutions to those challenges that we're facing as researchers. So just to echo what Mike said, we have been phenotyping for 10,000 years. Actually, phenotyping was one of the key things that came out of the Neolithic revolution as we domesticated species. So as a dog lover and as a connoisseur and occasional home brewer of beer, fermentation and domestication of animals also came out of the Neolithic revolution. So I feel like dog lovers, beer brewing, and phenotyping should all coincide as an appreciation for what Neolithic man gave us today. But you know, it's really impressive when we look at the history of phenotyping over the last 9,000 years, when you consider what teosinte is and what it has become with field corn today. But what's even more impressive is that idea of speed and how that has accelerated in the last 50 to 75 years. So if you look at breeding gains in corn, for example, if you look at where we were back in around 1920, before the advent of hybridization in corn, it took a land area about the equivalent of a half court of a basketball court to produce 1 bushel of corn. Through hybridization and impressive gains in yield through breeding, and then with the advent of biotechnology in the late 90s, which really has done a lot to protect those breeding gains on corn acreage. Today, we are at about a land area equivalent for a bushel of corn that is equivalent to the space within the free throw lane. So between the free throw line and the out of bounds, that's about how much land area it takes to produce a bushel of corn today. So think about that reduction in space and what that's, and the impact that's the positive impact that's really had on productivity and ability to use land as we feed a growing population. And we're projected to get produce an equivalent bushel on even less acreage as we move forward. So Mendel is considered the father of genetics. I think he's also foundational in phenotyping. And we can really trace our ideas of phenotyping and features that we're characterizing in plant growth and development back to those observational studies that Mendel had with peas. And everything that he was looking at genetically, he was describing as a phenotype. So phenotype is really critical for explaining genetic outcomes. And no matter how we modify the genome, the desired outcome always occurs at the level of the phenome. So whether you're looking at traditional breeding outcomes like Mendel was doing, or whether you're looking at non-Mendelian inheritance through epigenetics, or other types of modification through protoplast fusion or mutagenesis, transgenesis, polyploidy, and just recently there have been a number of products that have been released on the market that are the first generation products of gene editing. So the Browning-resistant mushrooms is one example of that. So no matter how we modify the genome, we always want to see what's happening at the level of the phenome. And the ability to modify the genome, the precision with which we can modify the phenome, the cost of modifying the genome, these are all becoming more and more, these are all making genome modification more and more accessible to not only industry, but also plant biology researchers. And so the scale of genome modification is increasing. So this is just reflecting some graphs that I'm sure all of you have seen, over the recent decade. But the cost of sequencing actually is moving faster than the decreasing cost of computational power by Moore's law. So where $3 billion was spent to sequence the human genome in the late 90s, you can now do it for about 1,000, which is pretty incredible. And as genome sequencing costs go down, of course, the amount of sequencing data that we accumulate increases, so we're up to the hundreds of trillions of base pairs in GenBank today. And similarly, as the cost of sequencing goes down, adjacent technologies like gene synthesis are also coming down. So all of this is pointing to our ability to accelerate genome modification, and the real demand to meet that acceleration in genome modification with corresponding increases and advancements in phenomics technology. So I want to talk a little bit about that, and I put this slide together sort of to editorialize some ideas that I've been thinking about over the past 6 months since I've been with LemnaTec, and I've been talking to customers, and I've been talking to people in the US and Western Europe about about sort of the state of where academia and industry are with phenotyping. And I tried to do sort of a manual spot fiery type view of my thinking on this. But I want to first talk about the size of these bubbles that I'm showing on this slide. And the size is, what I've done is really kind of taken size to represent utility. And one of the things that I hear a lot when I talk to people about phenotyping is a lot of interest. Almost one of the first things that come up are interest areas in these smaller bubbles, the NIR, infrared, chlorophyll fluorescence, fluorescence in general as a phenotyping technology. But what I will say is, in my experience and in what real world need really is, about 80 to 85% of the questions that you ask will rely on high resolution, reliable, repeatable, visible light imaging. That's always going to be the cornerstone of your phenotyping inquiry. There have been some really impressive advances in technology and capabilities in the last decade or two. 3D laser scanning is another really useful tool for phenotyping. It uses a laser-based approach to give you a very detailed three-dimensional reconstruction of the plants that you're using. And I'll talk about this now. I may repeat it again later, but one of the really powerful things that 3D laser scanning brings to phenotyping is its ability to bring information forward from some of these other systems. So at LemnaTec, we use a data analytical approach called sensor fusion, where we take the 3D image of a 3D laser scan and overlay it on the 2D images that are produced through, for example, a chlorophyll fluorescence system or an NIR system. And what that does is it corrects for many of the angle effects and confusion, frankly, in the data that you get from a strictly 2D image alone from some of these other modalities. So 3D laser scanning has a very high utility. And then finally, hyperspectral is just, there's a lot of really exciting things happening with hyperspectral imaging. So from field scanning, using hyperspectral to do scouting, early detection of disease, as well as taking it into the laboratory context. So there's a group out of the University of Bonn who's using hyperspectral imaging to study metabolic and other chemical compositional changes in plants. So there's a lot of real discovery phase things that are happening with hyperspectral imaging these days. So that's the size of the bubble. And then as you're thinking about cost, and this is all very subjective, so working in tens of thousands and working in orders of magnitude, not specific numbers here, but in terms of cost, visible light phenotyping is by far going to be sort of your entry-level solution for coming in the $5,000 to $20,000 range for very nice instrumentation. Then laser scanning, you start getting up into the $10,000 to $70,000 range, and it's very easy to spend over $100,000 on high-end hyperspectral cameras. So just some things to keep in mind as you're thinking about developing phenotyping solutions. I think more than anything, think about the kinds of questions that you're asking now, and think about the growth potential of the system. So how you can build on it as your needs change going forward. So I'm going to take this and take some of these ideas and talk about, I think just given the time, I'll probably talk a little bit about visible light imaging, how we're using it and some of the tools we've developed at LemnaTec, show you how we're using laser scanning, and then talk about a new instrument that we've developed for hyperspectral imaging, and then we'll see where we are and jump to the end. So, as I said, visible light imaging is really the cornerstone of any phenotyping system that you put in place. Visible light imaging is going to deliver the information that you need for plant size, plant architecture, color, and if it's a very high quality system, you can get a very high degree of temporal resolution, so you can start looking at change over time. And then you can use analytical solutions that I think Chris will talk to you more about to do things like looking at skeletonization to define leaf number, leaf length, angles, stem length, elongation rates, change over time. You can use color to quantify very specific features, so you can get an early view of damage, either due to herbicide or disease or abiotic stress, whatever your question is, using color. And you can correlate some of those color changes with specific responses. So, for example, you get a very different color response in Arabidopsis under nitrogen stress than you do drought stress. You can extend it further beyond plants to insect feeding assays. So this is something that a number of our industrial partners use our systems for. They use it for insect feeding assays on dietary plates or even leaf discs. And then finally, you can use visible light imaging to get a lot of information around post-harvest questions. So looking at things like at ear metrics, tip void, kernel, doing an estimate of total kernel count, individual seed metrics. You can get information about color and some compositional features based on how color changes over the dimensions of the seed. So essentially, all of our systems, whether it's a laboratory field or a greenhouse system, are all going to have visible light imaging as the foundation of the instrumentation. I'm just showing 3 examples of some of our instruments here. So on the left here, this is a manual individual plant phenotyping system. Actually, you can also do flats in the top-down version of this instrument, so you could do multiple plants. But it's essentially used. It's an entry-level system that's used for low-throughput phenotyping of individual flats or plants. It's a standalone system with touch screen control that also allows you to do on-the-fly data visualization, and it's available, as I'm showing here, for top-down view. But we also have a short wide one that you can use for side view. So if you're looking at taller plants, and especially for monocots, often you want to have that side view imaging. Up here at the top is a new system that we're developing this year. It's really kind of, I think of it as sort of as an entry level automated phenotyping solution. So you can buy the base unit that you see here with only visible light imaging, and you can start immediately doing high throughput phenotyping of flats of plants or microtiter plates or whatever your research area calls for. And you can then, it's modular in the sense that you can start adding on a tray feeder if your throughput increases. You can also add on other imaging units if your imaging demands increase, so that you can start expanding beyond visible light into things like chlorophyll fluorescence or hyperspectral. And then finally down here is just a picture of a conveyor system. Again, visible light is part of all of those systems, and these systems are larger greenhouse installations that have fully integrated growth and plant growth and maintenance and watering and so forth in them. 3D laser scanning, the way this works is you use a laser to generate a point cloud. And so those point clouds identify very specific features, and you can do this at a high degree of resolution and use machine learning type approaches to take those features that you're extracting via the point clouds and start using machine learning to identify specific organ level features, panicles, leaves, things like that on the plants. We have, let's see. So I just want to give you an example of how we're using 3D laser scanning in our field scanalizer system. So this is a system that is at the University of Arizona Maricopa field site, and this is part of the Terra reference project. And a major focus of this project is accelerating breeding in bioenergy sorghum to produce bioenergy sorghum as a commodity crop for the southeastern region of the United States. And we have many sensors within the sensor box that we use at the Maricopa site. Laser scanners are an important part of this, and as I said before, what the laser scanners allow us to do is that sensor fusion approach by coupling the 3D view that we generate from laser scanning with the 2D views of infrared, PS2, and hyperspec. So what I just want to show here is a view of what that looks like. So this is data that was actually captured over time from the Maricopa field site. And there's a couple of things that I want to point out with this. First of all, we're getting to the point with laser scanning and 3D imaging with laser scanning that from a Gantry system, we can singulate plants. So this was over time, and we were able to monitor the growth of these sets of plants individually over time. Now, the other thing that you can see is the lasers, the 3D images give you you can start seeing very distinct architectural differences between these two plots. So let's just focus on an easy one. You can see that there's very different panicle density between the one on the left and the one on the right, so that you can start identifying very specific features. One of the things that we're working on is to try to get laser scanning below, trying to get further down into the canopy. So while those are two of the strengths, one of the drawbacks is with 3D laser scanning from a gantry, you really get only the top half to third of the plant in that 3D view. So I won't spend too much time talking about specific indices with hyperspectral imaging, except to say that there are a wide variety of indices that are being developed using hyperspectral imaging. Everything from NDVI for remote imaging of fields and forests to specific indices that measure things like water, chlorophyll, and stress pigment composition. So there's a lot of discovery potential with hyperspec, and I think that there's going to be a lot more knowledge to be gained using hyperspec in the years ahead. We've recently developed a robotic unit that will allow us to use hyperspectral combined with the 3D laser to translate very complex plant structural level information into a very high resolution 3D model. And so this is a robot. It's got an articulating arm that's got hyperspectral 3D laser scanner, and then two lights that focus into a line, and you're basically doing all of your imaging in a line across the plant. So if the microphone here is the plant, you can very easily scan up with the robot, and then the articulating arm comes down, and it generates a very high resolution in 3 dimensions that way. I think that I'm going to jump through these next couple of slides, because there was one point that I thought was worth talking about with this audience, and I'm coming up on time here. So LemnaTec produces, when we're looking at greenhouse level solutions for phenotyping, LemnaTec really delivers greenhouse phenotyping solutions based on that are tailored to a research institution's demands. And so at the Nebraska Innovation Campus, we have a conveyor-based greenhouse system, and we recently completed at the Cranfield Agricultural Engineering Precision Innovation Center a gantry-based system. And there are a number of pros and cons to either of these systems that I think are worth evaluating as you're thinking about building out infrastructure in this way. So conveyor-based systems have been around for quite a number of years, and in many respects they're a very tried and true way of doing high throughput phenotyping. But some of the drawbacks of a conveyor system are that you have lower size and weight limits because of the intrinsic limits of the conveyor and also the intrinsic limits of the cabinets that you're bringing the plants through. In contrast, a gantry system, because the plants don't have to move, you're bringing the sensor to the plants. In a gantry system, you can basically phenotype large plants all the way out to maturity. So if you're asking questions of corn out to V7, then maybe a conveyor system is what you want. But if you really want to start asking reproductive stage questions around corn in a controlled environment, a gantry system is probably the solution that you're looking for. Conveyor systems, as I said, when we were talking about 3D imaging with the gantry, conveyor systems will give you high resolution whole plant imaging. The gantry system will be relying on 3D reconstructions, and so you're getting down to about halfway down the plant, but you won't be able to get all the way down to the pot. Now there are some solutions that you could consider with sort of imaging within the rows of a gantry system to supplement your gantry, but from the gantry itself, you're not going to be able to get the entire plant. However, with a conveyor system, while you're doing plant level tracking and analysis, which can be very important for basic research, the gantry system will allow you to do canopy level analysis. So if you're asking for how things translate more into a field context, the gantry system may be the way to go. The conveyor system, as I'm trying to communicate down here with this graph, is a fixed configuration. So there is some variation in how you can populate the greenhouse with a conveyor system, but in general, your pots are always going to be this far apart. So you could populate it with every other pot, or you can change your pot so that there's multiple plants within a carrier, but they're always going to be in this configuration that's defined by the conveyor. A gantry system, it's wide open, so you can configure the floor of the greenhouse however you want with a gantry. So there's a lot of opportunity there. However, with the conveyor system, the movement really mitigates any environmental variation that will occur in any greenhouse. So no matter how good your greenhouse is, there's always going to be environmental variation, and you'll need to take that into consideration with your experimental design with a gantry system. And yeah, one other real quick point. In cases where moving your plant can influence the outcome of the phenotype, obviously a gantry system is going to be a better choice. So a real clear example of that is thermal imaging. If you want to do anything with thermal imaging, as soon as you move the plant on a conveyor, you immediately disrupt your ability to do that in a biologically relevant way. You'll get data, but whether it's interpretable or meaningful. So we provide all of the software for all of the instrumentation that I just described, and I also want to give a shout out here to the Plant CV software, which really it's an open source software solution that overlaps a lot with some of the things that we do with LemnaGrid, and there's a lot of really exciting things happening with Plant CV these days in the plant community. I think I'll jump to the end here and just open it up to some final questions. So just to summarize, phenotyping really follows on genotyping. It's crucial that we develop tools to address this challenge as we become more capable in our ability to genotype and make genome modifications. And LemnaTec delivers solutions at a variety of scales, laboratory, greenhouse, and fields to do just that. So with that, I'll finish up.
Anyway, thanks for that talk. I enjoyed it. Kind of a nuts and bolts question. I like that robotic arm. And is it that the plant is also rotating, or is it that the robotic arm with the imaging equipment moves around the plant?
Yeah, the robot moves around the plant. So there's not a need for the plant to rotate that way. Now, in the cabinets that I showed on the conveyor system, actually, if you looked at that particular image, as the plant moves into those cabinets, it moves onto a stage that it lifts it, and that rotates. So then you can, from fixed cameras, you can get multiple angles. You can get a variety of different angles.
If you have several cameras, it's not like you have one and it kind of moves around.
So on the robot, there will be this is the plant. This is the robot. On the robot, there will be one camera that moves around, like that. In the cabinets, there's 2 cameras, maybe 3, but the plant moves. So in the cabinet configuration. So that's a higher throughput. So that kind of solution in the cabinet where you have, that's a higher throughput, lower resolution solution to the problem. The robot is specifically a solution we're developing. It's lower throughput but higher resolution. So,
what's the time per plant?
On the robot.
Yeah.
Well, it'll depend on the question you're asking, but you can easily test a greenhouse. If you take a conveyor greenhouse, you can easily test a greenhouse of 500 or even 1,000 or 2,000 plants in a day through visible light imaging. With the robotic unit, that's going to be like 5 to 10. That can be up. Hyperspec slows everything down. No matter whether it's on a robot or it's fixed mounted. Hyperspec slows everything down because you're looking at multiple you're capturing multiple wavelengths. That is probably going to be 5 to 10 minutes per plant, depending on the type of scan that you do. So, but you can get high throughput. You can test an entire greenhouse.
And let's thank Todd. Oh, sorry, there. Yeah.
Just a quick question.
Sure.
I see that LemnaTec is a growing company. You've been hiring a lot of people recently based on all these installations. So, from an educational standpoint, as we train graduate students, what are the skills that your company most values, and what skills are lacking? Do you see in people that are?
Great question. That's a really great question. So, the data analytics piece, developing the computational tools for handling the massive amounts of information that we're generating. So that Gantry system, Chris might actually be able to answer better, but it's generating terabytes of data per day. Yeah, I mean, it's just an enormous. So the data analytics piece, there's really important to be able to take the biological know-how and bridge it with the instrumentation. That's a really crucial piece. So understanding what the strengths, weaknesses, limits are of the sensor instrumentation that's out there is really crucial. And I'll think about that some more and be ready to talk to grad students at the break. It's a great question.
This has been a production of Cornell University, on the web at cornell.edu.